graduate fellows who are presenting their talks are Cassie, KaʻApu-Lyons, and Emily Conklin. Their talk is titled Fish Food for Thought: What do baby fish eat and why it matters? Give it up for Cassie and Emily.
Thank you, Maya. So we're not at the beginning of our slideshow here, but I'm Emily Conklin. This is Cassie. Hi. Perfect. There we go. And we're going to be talking today about fish food, what baby fish eat, and why it matters. Sorry, folks, technical difficulties. Oh, looks like it's working. Okay, thank you so much. Okay, our journey today begins with a trip to the local pet store to look at the aquarium fishes. So over 11 million households in the US have a home aquarium. It is a lot of people's first experience with tropical fishes. It helps them learn about the environment and nature and helps foster a sense of responsibility. So when we enter our pet store, we're drawn to the colorful walls of fishes, and we're first going to check out the freshwater selection.
So here we have our 13 most popular freshwater aquarium fishes. They include things like your plico, your tetras, and your fancy guppies. And 90% of freshwater fishes are raised in captivity and readily available. And we can show this by the yellow dots at the top of the screen, or at the top of each aquarium. Alternatively, if we look at the marine fish, only 3 are bred in captivity, and these include things like your yellow tang and your clownfishes. However, the other 10 are wild caught. Wild caught means they have been removed from the reefs. And although this can be done both safely and sustainably, harmful methods in the past have led to some concern by environmentalists and conservationists. And so there has been recently requests to ban collection of aquaculture or of aquarium fishes around the world.
So these potential closures highlight the mindset that aquaculture represents an important part of the marine aquarium industry moving forward. So some benefits of captive breeding include healthier fish. Fish raised in captivity can be reared to be disease-free. They adapt better to life in tanks, and they miss the stressful journey from reef to retail. It also provides job opportunities. So culturing marine ornamental fish is a $5 billion opportunity. And of course, it can help reduce fishing pressure on our wild reef stock. But getting the fish to reproduce in captivity is only half the battle. So if we return to our 13 most popular marine aquarium species, many of these species have been successfully bred in captivity, but can't be produced in commercial numbers. And overall, about 400 species of marine fish have been bred in captivity, but only about 50 of those are commonly available in pet stores.
Oops. Sorry. Reef fishes have a complicated life history. After they hatch from eggs, they can spend weeks to months as larvae in the open ocean before settling on a reef to call home. They're also very different from their adult counterparts. Larval fish occupy different habitats and have different diets. So this makes it complicated when we're trying to grow these up in captivity because we're unable to complete these life cycles. High mortality is often a result of disease and starvation. And while disease can be remediated and controlled, starvation is a bit more difficult, likely due to the fact that we don't know what these fish are eating. And so for our project, we're asking, what do larval fish eat in the wild? Because of their small size, smaller than your pinky nail, and scattered presence in the open ocean, larval fish are difficult to observe eating in the wild, and so we have to capture them and bring them into the lab. Then we homogenize them or blend them up. We extract the DNA. We put it into a machine that generates millions of copies of targeted DNA, and then run it through a sequencer that puts it into a language that we can interpret. And from there, we get different unique barcodes that have to be separated.
So these DNA sequences we get back are unique to specific organisms. So just like barcodes link products to prices at the supermarket, each DNA sequence can be linked to the organism it came from. So if we get back a mystery sequence from our fish's diet, we can use our barcode databases to try and figure out what that fish ate. So in this case, we can see that this fish ate this little shrimp down at the bottom right. So barcode databases are kind of like DNA dictionaries in which each species can be matched up to its specific sequence. So there are a lot of these databases publicly available online. Some of the ones we use are Barcode of Life and NCBI's GenBank databases.
The benefit to this technology is that we can look at all this potential species in the diet of a larval fish, as well as look at multiple species at the same time. And so to visualize, this plot is to help us visualize it, and it's a lot like a diet journal. Does the fish in red, so the fish species are on the right. Yeah, okay. Does the fish, yes, the left, sorry.
All right.
My right. And the prey items are on the right. And so do fish eat a lot of species, like the species indicated in red, or do they select a few, like the species in blue? We can also look at relationships between those fish species. Do they all eat the same types of prey items, or do some focus on just a few?
So our goal for this project is to apply this advanced genetic technology to asking what baby fish eat in the wild. So once we identify these diets, we can help set the table for aquaculture. We can work with growers to identify nutritional requirements for some of these difficult to grow species. And this has the potential to diversify and enhance the industry.
We aim to push aquaculture into the future. Not every fish and prey item is currently culturable or inexpensive, but we hope that with our data that we can help grow out these species to be enjoyed both in aquariums and on the reef. And with that, we'd like to thank all the many people who have helped us throughout this project and our funding sources, and Frank Ponch for all the larval fish pictures. And thank you guys for listening.
Thanks, Cassie and Emily. Questions for our researchers or scientists tonight? Yes.
So once you identify what the larval fish eat, is there going to be, I guess, this process again for how to grow those species that they eat?
Yes, there will be a next step. But it's not quite figured out from at that point. Yeah, hopefully I'll graduate by then.
Other questions for our scientists? Yes, Sarah.
I'm wondering how difficult it is to get enough. Can you repeat the question for our online audience? Or I can repeat it.
Go for it.
Okay, online Zoom audience, and you gotta stand close to me, Cassie, because you have one of these nice online mics. The question was, how difficult is it to get DNA out of a teeny tiny baby fish?
Okay.
Is that the question? Yes.
It hasn't proven to be difficult yet. I do get occasionally a couple empty stomachs, but I'm not sure if they're just empty stomachs or if it's been a problem with just the difficulty in the techniques. Yeah.
We'll take one more question from Shimi.
Or we'll take both questions. Let's do it. Let's go Shimi and then this question in the middle. What's your question?
What do you think it'll take to have people, because it's kind of a sport, right? Like it's just, because people love to, it's a high price to buy the fish that's wild caught. So like, yes, you know, like what does it take to stop people from catching? Like providing these alternatives, but then there's like a price. Like, how does it catch up to the price?
How does it catch up to the
Like,
price
how
of
does
it?
the growth of, you know, these baby fish so that like the fish can be bred in captivity versus like people thinking like, yeah, I'm gonna catch all this fish because I'm gonna go out and catch, you know, like how do you counteract that mindset of sport fishing almost, you know?
Online audience, the question's, how do you counter the mentality of sport fishermen, which is get as many as I possibly can?
Okay, so I think it really is going to end up being with these bans that they're proposing, because especially here, right? We kind of have that right to catch, and people are upset with it, but I think by doing that, we're kind of pushing aquaculture as being the future. So yeah, yeah.
This will be our last question.
So the question was, is there any relationship between salmon aquaculture and then this type of aquaculture? Well, like you said, salmon is very well established. I think for salmon, they did a lot of food trials. This is fairly new technology. And one of the reasons why this comes up is they've tried food trials, and a lot of the reasons why these fish can't be bred in captivity is because they're not able to feed the same things as a salmon. Each species seems to have a very specific diet, whether it's, you know, copepod, but one copepod moves a little bit differently than another species of copepod, or just a preference in krill versus copepods. I think they've gotten salmon diets pretty well honed in. Does that answer your question?
Okay, great questions. Another round of applause for Emily. Our next speaker is Patrick Nichols. His talk is titled, "It's Here, Now What? New Tools for Monitoring Marine Invaders." And while Patrick gets his lavalier microphone on, I want to remind everyone, closing night for the resiliency exhibit is this Friday, and we are going to screen a Voice of the Sea episode on Limu. So if you love Limu, come back on Friday. It's an early screening before it's premiering on TV. Let's bring Patrick up to the microphone.
Thank you, Maya, and appreciate all of you being here tonight. I'm going to talk briefly about a project that I've been working on, specifically developing a new tool to track the spread of invasive species. but first I'd like to introduce myself formally. That's me when I had more hair. My name is Patrick Nichols. I'm in the zoology graduate program at the University of Hawaiʻi at Manoa. I've always been extremely interested in the ocean. I could just sit there and look at it literally all day. I started my degree in marine biology, studying invasive species, most notably the Indo-Pacific lionfish, which was actually introduced into the Caribbean back in the late 80s at a single point in South Florida. And this was a single introduction. It has since spread throughout the entire Caribbean region. The spread has kind of gotten out of control at this point. It's even down past Brazil. So this map is a little outdated. As you can imagine, because it's such a voracious predator and establishes extremely quickly, it's had a negative impact on a lot of the native species in the region. And if you had known exactly where it was introduced back in the late 80s, it would have been much easier to eradicate the species. But now that it's so widespread, it's here to stay. Rather, management initiatives have focused instead on lowering the number of individuals by hunting them. And that's all they can do at this point to minimize their impact on the environment. So shifting gears a little bit closer to home, yet still very far away, Papahanaumokuakea Marine National Monument, located in the northwest Hawaiian Islands, is one of the largest marine reserves in the world. Its reefs and atolls are in near pristine condition, and they support diverse communities of reef fish, corals, and seabirds. However, its remoteness means that it is lacking in a lot of outside impacts that we have here on the reefs in the main Hawaiian Islands. Although some of the main threats to Papahanaumokuakea are actually sea level rise, marine debris, and invasive species. It's situated in a weird confluence of ocean currents that causes a lot of debris of all sorts to accumulate on the shores here. And in 2016, during a routine marine debris removal and reef surveys, researchers actually discovered a new species of algae at Manawai Atoll here in red. Now, this would have been extremely exciting to find a new species, but to the dismay of the people doing this research, this new species was found covering entire stretches of coral reefs. It was brought back to the lab and determined to be a new species named Chondria tumulosa, from the Latin of many mounds, given how many mounds it was forming along the reef. By 3 years later, it had spread across the entire atoll, and again, at many sites, smothering completely whatever native species were living under this dense mat of algae. And while we don't know exactly where it came from, it's likely that it was brought in, washed in here, or has been here all along and only recently started acting invasively under new conditions. Regardless, this was kind of alarming to the researchers. And given the limited resources and number of staff at Papahanaumokuakea, getting ahead of this spread meant that we needed to do more research on the exact extent of how far it has spread throughout the reserve. To make matters worse, the pandemic actually delayed and all the future expeditions up to the reserve and the monument to collect more data, which severely limited our impact and ability to collect more data on this species. So it's here, it's in the Northwest Hawaiian Islands. Now what do we do? It was around this time that I was teaching an undergraduate internship course at the university, where we placed students with local partners doing research in marine science and developing their skills. One of these partners was U.S. Fish and Wildlife Services. Dr. Kaua, pictured here speaking with our students, had heard about a technique that I was developing for my master's degree. What I was doing was I was essentially taking DNA from seawater, specifically for corals. And by looking at the DNA from corals, we were able to tell exactly what species were in any given area as well as their relative abundance on the reef. So US Fish and Wildlife was extremely interested in developing this technique to detect their algal invader. Now, it's actually quite complicated, but I'm going to just make it real brief here. I like to think of the process kind of as a crime scene, and I'm the forensics investigator. What I'm trying to do is determine, based on the DNA fingerprint in the water, whether or not this algae of interest was at our crime scene. And again, it's really quite simple. We collect DNA from the water. All living things give off DNA into the water. So we collect this DNA, and I developed a marker that specifically targets our algal invader, essentially isolating its DNA from everything else. We then run this through a fluorescence machine, which turns the amount of DNA in our samples into an actual positive detection that we can analyze and say exactly which samples had our chondria DNA in it. Now, using traditional techniques of visually surveying a reef, it would take a team of trained scientists months to survey the entire atoll, not to mention the entire reserve, being the largest, one of the largest in the world. However, using this new tool, we can survey sea surface water samples, which anybody can collect, and is extremely efficient. And because chondria is giving off DNA in the water, we can accurately detect where it is throughout the reserve. It's even more sensitive in certain instances, being able to detect chondria at extremely low abundances where divers and snorkelers would have otherwise missed. This was perfect timing because just recently, chondria tumulosa was discovered at neighboring Kauai or Midway Atoll, which is kind of an alarming step for this invader. So we're now using this tool to survey other neighboring atolls, as well as ports and harbors throughout the main Hawaiian Islands, because we suspect that if it's going to invade here, it would be from shipping. And the last thing we want to do is bring this invader with us from ships going to and from the reserve. Ideally, this tool can help our reefs looking like this rather than looking like this. Now, I must admit, it's kind of difficult working on this project personally because I've never actually seen Papahanaumokuakea. And admittedly, I've never even seen chondria tumulosa in person. I was brought on this project to develop the lab techniques. And while I do enjoy working in the lab, it's extremely easy to kind of forget what it means in the big picture for this whole story. However, we find ourselves at this kind of unique crossroads of being able to limit the spread of this invasive species early on in its establishment, which is actually kind of rare for any invasive species. And it usually seems like conservation measures are generally a couple steps behind what's actually needed. But this is actually one instance where I feel that this new technology can give managers a leg up and an advantage in order to actually make a substantial impact. If we do our job correctly, many of you may never hear of chondria tumulosa again. However, more realistically, it's here to stay, but we can use technology, this new technology, to minimize its impact. And it's pretty unbelievable to think of the immense amount of time and effort it takes managers to maintain these pristine areas, some of the most remote in the entire world. It took a community of all around dedicated individuals working day and night and some pretty insane logistics. I wouldn't actually know I wasn't there, but shout out to Randy and the audience here. Big help on this project as well. And all of our collaborators we've had from multiple government agencies, as well as local nonprofits and research institutions from near and far, all offering their expertise in different areas. Couldn't have been done without all of these people. And all these collaborators care deeply about what's going on up there in order to dedicate their time and resources to this massive effort. And I'm extremely grateful to be part of that in order to give a little bit of my expertise to help protect the diversity of our reefs. So that's all I have for this evening. If anybody has any questions, I'd be happy to answer them.
Holo, questions for Patrick? Let's see, one for Rosie and then from Pat.
Do you have any idea where it came from? Like, you know, the introduction, like, let's see how origin source.
So yeah, the question was, do we know where it came from, the origin source? So we're currently working on that, not me personally, but the movement is to go there next and see if we can determine where exactly it came from. There's been a couple ideas, again, that one, it's kind of been here all along at low abundance, and only because of climate change or shifting conditions, it now has bloomed into this issue. It could also have come from ocean currents, again, like Johnston Atoll, or one of those stepping stones across the Pacific.
Al, did you have a question?
I was just curious, do you have to take into account the circulation of the water in making these samples?
Yeah, good question. So the question was, do we have to take into account ocean currents and movement of the water when we're taking our samples? And yes, so we generally have to take down all the metadata of what's happening, how much waves there are, how windy it is, the tide level, that sort of thing, because all of that can move DNA through the water. So just because we find DNA in our samples doesn't necessarily mean that chondria was right there. But we found actually, incidentally, that it is extremely accurate, and on the order of, in this room, I could tell if chondria was here. So that's kind of the scale that we're looking at.
Another round of applause for Patrick. Give it up for Patrick.
You're back.
Our third speaker tonight is Sheldon Rosa, and his talk is titled Holo, Anaeholo, Anaeholo.
Welcome, KaʻApu. I'll be making some time to come down to this presentation. Today, we're going to be presenting a little bit about one of our main species that we're researching at the fish pond. Oh, the thing going by itself. But aloha, my name is Sheldon Rosa. I'm a master's student in the oceanography program at UH Manoa, and going to be talking about the Anaeholo. So the Anaeholo is also known as the Hawaiian striped mullet, or Magocephalus for the scientific name. It goes by a bunch of different Hawaiian names as well. Ka'i'a ka'awawai is one of them. Ka'i'a is fish, ka'awawai means struck by your feet. So traditionally in some areas it was struck, people would run to the shores, kick it with their feet, and then people would grab it on the shore, and then that's how they got their dinner. It also has names based off of its growth sizes. So if everybody puts out their finger, anything in this general area is called pua'ama. Everybody say pua'ama. Oh, you guys good already. Then if we put out our hand, from the base of it to our fingertip, this is kahaha. Everybody say kahaha. All right. And then from our elbow to our fingertips, this is ama ama. Everybody say ama ama. And then anything bigger than this is anai. Everybody say anai. All right. So we're going to be speaking about the anai. These are our mature mullets. And this Anaeholo that we're talking about are talking about the schooling mullets that happen and they go around Oahu Island. And then anaipali is another term that goes with it. The importance of this species is that it's a delicacy within a lot of communities. Here's a song by Sam Alama called Ama Ama, talking about eating this fish steamed in tea leaf. And right after you eat it, you get the special kanaka tap, go sleep right after. So talking about the ono and the great, just how good this fish was. Mano lelo no ea, a proverb that goes with this Anaeholo, is ka'i'a hali akamakani, which is the fish fetched by the wind. So Anaeholos generally start on November months and ends in March and April. This is our last Anaeholo that we had in Waikiki. No, I'm just joking, this is in Florida. So the cool thing, Florida has the same mullets that we have in Hawaii. So they also have Magocephalus in their coastal waters, and they have the same kind of runs. So When we imagine it, this is kind of what we would be seeing if a full potential Anaeholo were to happen along our coastlines. And then there's a newspaper article from the Honolulu Advertiser in February 21st, 1939, talking about basically what we're seeing, huge schools of fish getting struck down by different predatory fish like sharks, swordfish, and ulua. So going into a mo'olelo about Anaeholo, this is in Ko'a Homerula. It's an English newspaper, but in the Hawaiian language newspaper article database published in December 11, 1908. And it talks about two couples. I'll just jump into the story. Don't take too much time on the summary, but there's two couples that were living in Laie. So the wahine of this couple, she had a brother in Kaʻihu-Pala'ai, or kind of West Lock area in Honouliuli. So the wahine in this couple in Laie, she's originally from this area by West Lock. So one day she gets a craving for this fish, and she kind of reminisces, and then she talks to her kane, or her husband, and she says, "Oh baby, you know that time when I lived, Honouliuli with my brother Iho Palai? Oh, we always had fish. I could always smell the tea leaf roasting, and there was always fat mullet on the table. But now I stay living with you. I get none of this right now. This is big pilikia. Get poi, but no fish." So when your wife says she has a problem, you have a big problem. So this Kane, this is the guy right here. He walks all the way from Laie, all the way to Eva to go talk to her brother. She gives him specific instructions. Make sure you go to my brother. Don't take the dry fish. Because how are you going to carry that all the way back to Laie? You're going to break your back. He's like, okay, I got you, honey. No worries. So he goes, he makes it. He meets his brother-in-law, Iho Pala'ai. And then another person in this thing is Ku'ula. He's a deity who takes care of these Anaeholos and is a fishing akua. So when he gets there, he meets his brother-in-law and he makes his request. Your sister, she liked fish, but no more fish on our side. So what do I do? And immediately, Ihu Palai brings out several bundles of the best dried fish you can think about. He's like, "Here, bro, I got you." And he's like, no, I cannot take that. She said not to take this. He's like, "Okay, what I'm going to do, I'm going to do in Pule to Ku'ula. And then what you got to do, go down the Kona Coast. Kona Coast is known as Townside right now. This is what it's, the moku of that. So go down to Kona Coast, go to Pu'uloa, go to Kumumanu, to Ko, which is Honolulu Harbor. Go to Kalia, Ala Moana Shopping Center. Go to Ka'alawai, we know where that is. Waialai, Wailupe, continue on, go around the Ko'olapoko coast to Makapu'u, continue up to Waimanalo. Keep going, do not stop, rest, sleep, nothing. Keep going until you see the eyes of your lover. And he's like, "Okay, not too bad, I think I can do that." So he makes his way, he goes to these different places, and as he's going, you guys can see that big white yellow line going. Never have cars back then. We can't even, 45 minutes in traffic is a little bit hard. So he's going, he's making his way, he's going up, and walking this far, you get tired. You're not supposed to stop, but he takes a break, he rests, and like, "Oh, okay, I'm tired, I take a break." And then he notices there's a big school of fish all on the shore. And when he's resting, he's noticing, he's like, "Oh, how come get all these guys running to the shore, grabbing all this fish?" He's like, "Oh, I wish I could go in, but the guy, my brother, told me not to stop." So he keeps going. And every time he does a stop, he notices this. Fishers are going, catching these fishes. And then he keeps on going and going all the way until he reaches Laie by Kaipapa'u to see his lover. When he gets there, he's a little bit mad because he walked from Evo all the way to Laie and he has no fish. So he goes back and he goes, "Oh, babes, bro, I went and walked all the way from Evo, all the way to Makapu'u, to Laie. That's what your brother told me. I have nothing. I don't know one tail, no eye of one fish, nothing." He was like, "Your brother, I don't know what's wrong with him. I don't know what I did." He's like, "Honey, no worries. You never see that school of fish following you?" He was like, "Oh yeah." He's like, "That is the fish that my brother went sent with you. Let's go down there and get some." So happy wife, happy life. So this is the story of the Anaeholo. Connecting to this story about why we want to continue this story, the Anaeholo, and how important this species is. In the 2008 Lawaiʻa magazine, there was an interview with one of the kupuna in 1976, and he talks about this being the last mullet run that he's experienced. And he actually talks about it going down the Waianae coast back to Honouliuli. And he kind of suggests and hints like the reason why he thinks that there's like not been strong Anaeholo is the degradation as well as the development of many of the shallow inland estuaries that we mentioned. So Kumumanu, Pu'uloa, Kalia, all of these different areas that were key spawning grounds for this fish. Another inquiry was the introduction of invasive fish. So tilapia being another one during that time. You know, we all see them in Ala Moana, Ka'alawai, all of these different areas. So this connects really to our project and how we want to connect all of our different local i'i'as together to track our main species. This is a very important species within local i'i'a, so we want to make sure we understand it, its life cycles, and also how our environmental factors and what we do on land impact it. So our goal is to figure out what the ama and the kondo mullet, which is a mullet from Marquesia, what they're eating in the pond and figuring out where they're inhabiting, specifically at local i'i'a or he'i'a. Just to learn more, create this better relationship between our fish species and our environment. So we're collaborating with a bunch of different people. Pai o He'i'a, big mahalo to Hi'ile and Keli'i for allowing us to do some awesome research with them. My partner Lani is also within this, and we're doing collaborative work with different people. Kua, Brenda folks, helping us to connect us with different local i'i'a, as well as the different faculty, my advisor, Rosie in the oceanography program for helping us through this research. PICAS and Sea Grant for helping with the funding, and the many hands at the nurse, Shini, Fred, Kavika, Eileen, Nali, for mentoring through this process, helping us mentor through this process. But that is the story. Pipi holo ka'au. Mahalo nui for listening. This is a video that I would like to share. This was in October 2020 of kind of drone footage at Kaimana Beach in Waikiki. Kind of showing a little ball of fish that occurred over there with different predatory fish whacking it. We don't know if this is mullet, but it's a cool coincidence to see, and we hope that one day we can see this grow and continue this story of the Anaeholo. But that's all. Mahalo. Thank you.
Wow, awesome. Thank you, Sheldon. Questions for Sheldon?
Sheldon, as you start to get into your research on mullet,
What are some of the kinds of things that you will be sampling? Are you taking water samples? Are you trying to find fish? What exactly are you guys sampling?
Question. And our researchers before kind of gave a really excellent explanation of what we're trying to do metabarcoding to find DNA within our sediments first, to see what kind of microbial communities are growing within this. And we're going to be doing mullet gut kind of sampling to see what's occurring in their guts. And we're also going to be doing amino acid analysis to track nitrogen and see where they're sitting on the trophic level. So those are the different kind of things that we're exploring.
Another round of applause for Sheldon. Thanks so much.
Our fourth and final talk for the night is going to be from Sadichchha Shrestha, and her talk is titled "When It Rains, It Pours: The Economic Impact of Severe Weather Events in Hawaii." Big round of applause for Sadichchha.
That the first slide?
No.
Oh,
There we go. That looks good.
sorry.
It's okay. Aloha, everyone. I'm Sadichchha, and I am a second-year PhD student in economics at the University of Hawaii at Manoa. And currently, I've been working on my Sea Grant research project with my advisor, Professor Peter Faleki, and other research team members, Professor McKenna Kaufman, Professor Nori Tarui, Dr. Victoria Keener, and a colleague of mine, Luke Miller, who was supposed to be here today, but unfortunately got sick. But yeah, I'd like to get started. So so first of all, the title of my talk is When It Rains, It Pours, the Economic Impacts of Severe Weather Events. So first, I'd just like to talk, define what extreme weather event really means. So as per the United States Department of Agriculture, extreme weather events or severe weather events means occurrences of severe weather or climatic conditions that causes are occurrences of unusually severe weather or climatic conditions that can cause devastating impacts on communities and agricultural and natural ecosystems. So some examples of extreme weather events are Manoa Valley flood of 2004. This happened in 2004, and this caused a damage of worth $85,000 US. So basically, the flood swept away a lot of valuable and precious manuscripts and documents from the Manoa Library at the university. We have the Hurricane Iniki from the year 1992. So this has to be the most powerful hurricane in the entire history of Hawaii, and it caused a damage of about $7.4 billion. The Kauai flood of 2018. So on the right, you'll see a picture of flooding at the intersection of Kuhio Highway and Wainiha Powerhouse. And on the left, you'll see a house destroyed at the Wainiha Powerhouse. So these are like some examples of extreme weather events that we want to focus on in our study. So let us talk about what this project is all about. So as I mentioned earlier, we're focusing on extreme weather events. So we're particularly looking at hydroclimatological weather events such as hurricanes, extreme rainfall, and wind events. And we're looking at different kind of economic activities. So when I think about economic activities, a lot of things come into my mind. It could be unemployment, it could be inflation, it could be exchange rates. A lot of things can be studied when you think about economic activities. So here are some examples. So for example, housing prices. So we know that real estate is a really big deal in Hawaii. It accounts for about 20% of the GDP of Hawaii. And so I think it'll be interesting to study what's going to happen if there's a flood, let's say in Manoa Valley. Like, are the housing prices within the Manoa Valley going to increase, going to decrease? I think that's going to be an interesting thing to look at. Nightlight intensities. So nightlights are captured by satellites and have been increasingly used by scholars, researchers, and economists to that serve as an economic proxy, that serves as proxy for economic activity. So for example, if you see on the top right, there is a picture of the US nightlight. So what it means is basically the higher the light, the higher the economic activity in that particular location. So we've been thinking of also looking at this. We've been thinking of looking at foot traffic, which is defined as the number of people that enter a mall, store, or a location. So for example, after a flooding event, is it more likely that people walk to the nearest store, the mart, or they just rather sit home and deliver and like get their stuff delivered at their home? And we might also be interested in looking at tourism, because tourism is a really important component of Hawaii. It accounts for about 18 to 20% of Hawaii's GDP. So we'd be interested in looking at what happens to the number of tourist arrivals, for example, in the aftermath of a severe weather event. So what do we really want to achieve? So through this research project, what we want to really do is we want to, first of all, be able to quantify the economic impacts of historical extreme weather events in Hawaiian economy. So I gave examples of 3 extreme weather events, such as the Hurricane Iniki, the Manoa Valley flood, and the Kauai flood of 2018. And what we want to do in our second step is basically use the quantifications or the estimates from the first step to be able to predict the impact of events of different scales, and taking into account the consideration of the frequency and intensity of such weather events in the future. So here's a simple illustration. So this is kind of like an approach that we're going to be using. So if you have two regions, region A and region B, and let's say that region A and B have like similar characteristics, but region A gets hit by a disaster, whereas region B does not get hit by a disaster. So as you see on the right side, because we're assuming that region A and region B have similar characteristics, we can basically extrapolate on the trend of activities for region A that gets hit by a disaster had there not been a disaster. And so the difference between the yellow dashed curve and the black curve will give you the difference of the causal impact of the disaster. So that's what we are trying to achieve. This is just a simple illustration. And so finally, talking about who will our project benefit. So we think that stakeholders and decision makers involved in climate change adaptation are definitely going to benefit from our project, because the results from our study will be used to inform the decision makers about the cost of taking no action. Cost of taking no action, a baseline against which planned interventions can be evaluated. So for example, it could serve to inform participation in new insurance markets or investment in critical infrastructures. So with that, I'd like to end my talk. Thank you.
Okay, awesome. Thank you, Sadichchha, for your talk. Any questions for Sadichchha? Sadichchha, remind us again, are you looking at specific events? So you showed us some photos from the flood in Manoa, the flood on Kauai, and Hurricane Iniki. How many of these different events are you going to look at in the study?
We want to look at as many events as possible, as many events as we can find. So right now we're just trying to narrow down all the events and have been trying to look at how much economic damages they've caused. So we'd like to look at as many as possible.
Oh, very cool. Okay, and last question of the night from Sheldon.
Within your guys' study, are you guys gonna look at kind of climate change, sea level rise impacts on top of it? For instance, for kind of looking back roads and other houses, how that would be played.
Can you repeat? Sorry.
The question is, are you going to look at the combined effects of the storm events and sea level rise?
Probably not. Our project is more focused on storm events. There's a different project going on that looks at the impact of sea level rise. So that's a different project, but this one is going to be specifically focused on storm events, although there is an intersection definitely.
Thank you so much. A big round of applause for all of our The last thing we're going to do tonight is maybe as you walked in, you picked up a raffle ticket. We're raffling off a copy of the Limu Eater tonight. So we're going to have Heather pick the lucky number from the lucky box. And as she picks the lucky number from the lucky box, we'll ask the audience for a drum roll, please. Oh my gosh. Okay, it's going to be the last three numbers on the ticket. And the winning raffle ticket tonight is 553. Does anyone have it? You got it? Oh, we have a winner in the back. Okay, congratulations. Okay. Thank you guys so much again for joining us for the Science by the Sea seminar. Hope to see some of you on Friday for our closing event for the resiliency exhibit. Enjoy the gallery and hope to see you on Friday. Thanks again.